So I'm going to talk today, I want to go over just some principles because I think that's really important, but just kind of blend in some main example diseases that I think are important to know about. If something doesn't make sense, because we got a whole hour and a half, I would prefer that you raised your hand and say, hey, can you explain that a little bit more? Can we talk about that a little bit more? Then get through the whole talk if there's something you really want to focus on. So basic, start with the basic concepts and facts. In order to have disease, there's 4 things that have to be present. You got to have the pathogen, you got to have a host that's susceptible to the pathogen, environment has to be favorable, and you got to have a little bit of time for the disease to develop. So when it comes to management, we look at trying to impact each and every one of those, particularly when it comes to biologically based organic management. We want to try to avoid the pathogen if we can, have a host that's not so susceptible, avoid favorable conditions, and avoid that time for the disease to develop. So sometimes you can get out of it, save a crop, avoid a disease problem if you just harvest it fast. One of the primary examples would be something like basal downy mildew. Basal you can harvest at any stage. So if that disease comes in, just be on the outlook, you catch it early, and just try to harvest and really push the crop. So foundation of management is knowing the pathogen, understanding its biology. And I think more so with an organic management program, that's what you've got to understand. And realizing that our pathogens vary a lot in how they survive, how they disperse, what their host range is. So not every pathogen can survive in the soil. So managing through rotation is not always going to be the answer for every single disease, and that's important to understand. Some pathogens can spread by wind really long distances. Others can only move within your field. That's important to understand. Some will have multiple sources of where they can come from. So you need to understand all those potential sources to have a good integrated management program. Types of pathogens. We've got a long list of what types of pathogens they are. Understanding the differences amongst them, what different types there are, I think is really important. Most of our diseases of plants are caused by fungi. These include diseases like the powdery mildews. We have another group that's called the oomycetes. They're similar to fungi. They used to be considered fungi. They aren't anymore. They're more closely related biologically to the blue-green algae. And these are the pathogens that cause things like late blight and the downy mildews. Next on the list are bacteria, one-celled organisms. They cause diseases like bacterial wilt, black rot of crucifers. Then viruses. So a number of these things are similar kind of organisms that cause diseases in us and in animals, but they're not the same organisms, thank goodness. So there are some parallels between the type of organisms that cause diseases in plants to what causes diseases in us and in other animals. And then we get down to things that we don't see as often as causing diseases in people and animals, such as the nematodes. There are some diseases in Africa caused by nematodes, but not in the US. And these are really tiny worms. There's some good nematodes and there's some bad nematodes. Parasitic higher plants, mistletoe and dodder. Other biotic organisms, we get into some really small, teeny things, even smaller than viruses that cause plant diseases. And then abiotic things get lumped in as being plant pathogens, such as air pollutants. So there's your long list of what can cause plant diseases. Some other just kind of key facts and things you need to know is with our fungal and our oomycete pathogens, those are the fungi-like ones, is they reproduce both sexually and asexually. And so when we're used to thinking of insects, we're thinking of things that have to reproduce sexually. Same thing with us and other animals. But these pathogens have a whole nother cycling, a way of reproducing themselves, which is asexual. And that means they can often reproduce really fast when they've got this asexual cycle, because they don't have to get together with another individual to make spores. They can just immediately produce spores by themselves, which means you can get into a cycle that's taking about a week. So about a week from a spore being blown from one leaf, landing on another leaf, to when you have another spot. So that can be pretty quick. The other thing that's important to realize is that these fungal and oomycete pathogens, there's several different biological types that you can have. They can be obligates, which means that they have to have living plant tissue to survive. Once the plant is dead, they're gone. There's some others are what we call non-obligates. They can survive in infested debris. Those are the ones that you can go after with the rotation because they're surviving the debris. Once the debris is gone, they're gone. So something like a powdery mildew that's an obligate, if it hasn't produced its sexual cycle, it can't survive in the soil, it's gone. Basal downy mildew, it's gone once the plant dies. But something else that can survive in debris, something like early blight, septorial leaf spot of tomatoes, bacterial diseases in tomatoes, they'll survive on the debris as long as the debris is there. Once you've rotated for a year or two and the debris is broken down and is gone, the pathogen's gone. And then we get things that we call soil inhabitants, and those are fungi that can survive in the soil without a pathogen. Without a host, rather, being present. So they can survive a while. They're really hard to manage through rotation.
Examples of those are?
Examples of our soil inhabitants are a lot of our root rotters can be soil inhabitants. So Pythium, Phytophthora, and they are our oomycete types, the Pythium and Phytophthora. Sources of pathogens, they vary a lot, and they differ from pathogen to pathogen, so that's really important to keep in mind. This is where you need to really understand the biology of the pathogen that you're trying to control. There's a lot of info on the web that you can find once you know what you're going after. So first and foremost, you can have plants in another location. If you've got a pathogen that's producing wind-dispersed spores, it may be another farm. It could be quite a distance from you with something like a powdery mildew or a downy mildew. And it can be dispersed long distances. And then we get some other pathogens that don't produce a spore that can move so far. A typical example is our Alternaria pathogens. They produce a larger, fatter, heavier spore. It just can't get lifted up by the air and moved as easily as a powdery mildew or a downy mildew. So they tend to be more localized and on your farm and not moving between farms quite as much. Alternate hosts. They can be weeds, they can be other crops. They can be sources of these wind-dispersed spores, and also some spores that are splash dispersed. Our bacteria are also splash dispersed, so they can get moved from another host, another crop, either on your farm or elsewhere. So they're really important things to be targeting when it comes to management. And that's where it helps to understand the host range of your pathogen. Can it be on weeds? Is it not on weeds? Insect vectors. Some of our pathogens are moved by insects, and that's going to be the way to manage them, is to go after those insects for management. Viruses tend to be moved by, most of them by aphids. Some viruses are moved by thrips, leafhoppers. It all depends upon the virus that you're going after. Bacterial wilt affecting cucurbits, that one is moved by the cucumber beetles. So that's how you're going to manage that one. So just as some examples. Infested seed. Some pathogens are able to be either on the seed or they're actually able to get inside of the seed. And that can be a really important source of some of these pathogens. Also a way that they can survive within your field. If you have volunteers that come up from those plants another year, that can be an important source. Give you an example, a really important example, the new disease basal downy mildew most likely came to the US on infested seed in 2007. So there's an example of a pathogen moving a really long distance in infested seed. Really important to have in mind what pathogens can be in seed if you're saving your own seed. And I know a lot of us growing organically like to try to save our own seed. Infested debris. Some pathogens can survive in debris, so that can be in the soil, that can be in your greenhouse from year to year. There's also a virus that can survive in tobacco. So if you're a smoker or any of your workers are smokers, this virus can be in tobacco. That's why the recommendation is to always wash hands after smoking. Survival structures. Some pathogens produce survival structures. Example is Sclerosis. I'll show you a picture next. And nematodes produce cysts. These structures, if you got a pathogen you're going after that produces these structures, they can survive a really long time in the soil, some of them. That makes it really hard to manage them with rotation. And then our soil inhabitants can live in the soil in the absence of hosts. And some of our pathogens can live on roots. So the Phytophthora that causes Phytophthora Blight in peppers and cucurbits, it can survive on the roots of purslane. So therefore it's important to manage that weed if you've had problems with Phytophthora Blight. Here are the Sclerosis I was just talking about. They can form on the top of some plants. So here it is with cabbage, called white mold. Pathogen produces these pea-like black structures. Here it is on beans, white mold. This pathogen has a really big host range. With, come on slide, with tomatoes and also peppers, it produces these things inside of the stems. So therefore, if you want to be looking when you're trying to diagnose a plant disease. So you want to look for structures on the plant, inside of the plant if you think you might have white mold. So with white mold, you get these white stems and then the structures form inside. And as I said, they can survive a really long time. So this is one, if you see it and you diagnose it, roguing out that plant tissue is a really good, important management step. Now, what you might think as you look at those structures, and I guess a step back here, is knowing that they survive in the soil, you might guess, oh, I bet the pathogen grows out from those structures. And this is where it helps to really understand the biology, because that's not what happens. What this pathogen does is it produces these little tiny mushroom-type things. Really is a fungus. This is what they look like in the soil. And from those, they release spores that disperse. So this pathogen is going to move longer distances than you might think from just looking at that little seed and thinking, okay, it germinates and grows from there. There's a really good biological control product called Contans, and what it is, that biological control thing, that fungus in that product attacks these sclerotia and actually eats them and destroys them. Dispersal mechanisms, we've touched on this, I've touched on this a little bit so far, but how do these pathogens get around? Wind, rain, also irrigation, so splashing, some of them really splash. Soil, this is soil-borne pathogen, it can move in soil. That's something to think about as you're working from field to field, that you want to clean equipment, or if you're sharing equipment with another farmer, you're going to want to clean. Seed for those pathogens that can be seed-borne. Insects for the ones that can be vectored by insects, and then yourself and your workers. You can move things around when you're handling plants. You can move things around on equipment. That's why one of the recommendations is not to work a field when plants are wet. Bacteria can be on the wetness of the leaves, and as you're working, you can pick it up and move it along as you go. So you want to wait until plants are dry if you've potentially got a bacterial disease in your field. So our control practices are going to focus back on understanding these issues. And I guess I should point out, as people are madly taking notes, I've got a lot of this information up on my website. I've got a section just on managing things biologically, or managing things organically. So a lot of this information is up there, and I'm continually trying to find time to get more and more info up there, so hopefully that will help you all out. So controlling the source, so start with pathogen-free seeds or plants. Make sure you inspect. If you're buying seed, ask, has it been tested for seed-borne pathogens? When you see diseased plant tissue, sometimes it can help to remove it, and that would be the case for a pathogen that's not being wind dispersed. So something like the tomato with white mold that I showed you, removing that plant tissue can really help. You got something like powdery mildew, it's a lot harder to achieve anything by trying to remove plant disease tissue. Minimizing the opportunity for dispersal. So our pathogens that are in the soil, if you cover that soil with a mulch, there's going to be less chance for the pathogen to get spread up and onto your plants. Watering at the base of the plant, so you're not splashing water. So using drip irrigation instead of overhead irrigation will help a lot. If you're stuck with using overhead irrigation, starting your irrigation when plants are dry and ending when you know there's going to be a good drying period will really help. And the reason for that is because that's going to reduce leaf wetness. Reducing plant susceptibility. When you've got the opportunity to work with, use a disease-resistant variety, that's a really good management practice. But otherwise, some plants, if they're a little stressed, they're going to be more susceptible to disease. But really realize also that if you've got a really healthy, luxuriant plant, sometimes it can be more susceptible to other diseases. And it varies amongst these diseases. So you want to have a plant that's at its optimum. It's not stressed, and it's not too luxuriant and growing too well. Making the environment less favorable for disease. Really important management step. And primarily what we're going after is trying to reduce leaf wetness and humidity. And the reason for this is a lot of our pathogens need leaf tissue to be wet in order to infect. A lot of our fungi need this, a lot of bacteria, they need a period of time when the leaf is wet so they can grow into the leaf and then they're protected. Some of our pathogens, particularly the downy mildews and late blight, they just need high humidity. Humidity above about 85, 90%, that's sufficient for them. They don't need free moisture on the leaf. So those are the kind of diseases you will see in a high tunnel. You won't be able to avoid with a high tunnel. Other pathogens that need to be splash dispersed, need wet leaves, you can control well with a high tunnel. So examples would be if you're trying to grow tomatoes in a high tunnel, you're much less likely to see early blight or septorial leaf spot unless you've got some condensation dripping issues in your high tunnel. But what you'll get into troubles with will be leaf mold, late blight can develop there, bacterial diseases will be less in a high tunnel. Other diseases that you can get into trouble with in a high tunnel with tomatoes would be powdery mildews. Powdery mildews do not need much humidity at all in order to infect. They're kind of our dry pathogens. So these are all the biology things you need to be thinking about when you're trying to manage things. And then soil moisture, we've got some pathogens that really like a wet soil. Our pythiums, our phytophtheras, we call them water molds because they really like wet soil conditions. So keeping the soil not too moist will help out a lot with those. You want to be looking at your plants all the time. Identify what the issue is. If you've got any problems with trying to figure out what you've got, there's a lot of pictures on the web now that will help you with identification, but realize that every state's got extension offices that can help you out with diagnosis. Make sure you've got it correct. I've seen issues of people thinking that they've got a really nasty disease like late blight and hauling out the plants and then realizing, oh, it was just drought when water stress symptoms on the leaves. So make sure you got it correctly diagnosed. If you're going to use an organic fungicide, you've got to start early if you're going to get control. With plant diseases, there's no cures. Insects, you can sometimes let the population sit for a little bit and try and decide, okay, is it at a point where I need to manage it or not? But with your plant pathogens, once they're inside leaf tissue, you can't stop those. All you're doing is trying to protect what leaf tissue isn't infected at this point in time. And I think that's something that's really important to keep in mind with plant diseases compared to diseases that affect, for instance, us. You can get really darn sick with the flu and you can recover. You can get measles and be spotted and you can recover. But a plant, you're never going to get rid of those leaf spots. So you got to keep that in mind. So what are some of our seed-borne diseases? There's a number that affect tomatoes. I think tomatoes is a really good one to focus a little bit on because I know a lot of people like to try to save their own seed with tomatoes. So we've got 3 bacterial diseases, canker spot and speck. Here's some symptoms of speck. Spot's kind of similar to that. Septorial leaf spot, where it's more of a circular spot and gray in the center. So how do the pathogens get there? Well, sometimes they can be systemic, but the other thing that happens with tomatoes is every once in a while the seed coat is stuck on the cotyledon. So if the pathogen happens to be on that seed, life is golden for the pathogen because it's right there on the leaf tissue. We've been using hot water seed treatment up in New England area for treating seed. You can do this on the stovetop with a big pot with careful control of the temperature. The best way to do it is with precision equipment. There is equipment here in Virginia. I know Steve Rideout was part of this project that I was involved with in getting this equipment. So he has equipment. So if anybody's interested in doing hot water treating of seed, tomatoes, eggplants, peppers, brassicas are all ones that handle hot water seed treatment well. And what we've got with this equipment is equipment that we can precisely hold the temperature. Because what you've got is we're treating the seed at a temperature that the seed can handle, so the highest temperature the seed can handle without affecting germination, but high enough so that it will kill the pathogen inside the seed. And then a precise amount of time that the treatment's going on for. Tomato in particular holds up very well to this. And sometimes if you treat the seed soon before you plant it, you can get kind of a priming effect on the seeds. You can get improved germination. If you hold the seed for too long, so if you treat seed, plant some this year and you hold it for a year or two, you definitely might see some reductions in germination. Some companies will hot water treat seed, so that's something to check on, whether or not the company you're working with will do it for you. Here's another pathogen that can be inside of seed. This is a bacterial pathogen. This actually gets inside of the veins of the leaves. This is black rot, so it hits cabbage, cauliflower, kale. I've seen a lot of issues with kale. We had a lot of problems last year with it in kale. I don't know if any of you had issues with it in kale last year. So the typical symptom is these V-shaped lesions around the edge of the leaf. And if you look really closely, you'll see associated with that lesion is these black veins. And that's because the pathogen is inside of these veins and it's clogging them up, and that's where, you know, the tissue's dying, you get this blackness. You can also see systemic symptoms, and these symptoms are from the bacteria moving up, and that's probably more from a systemic infection. So that's, if the seed is infected, you'll get those kind of symptoms. So just to give you a little biology with this pathogen, so here it is, it's in the veins, and what will happen is, you know how overnight you get guttation droplets on the edge of leaves? That's water that's coming out of these veins. So you can imagine if water's coming out of these veins, what's going to be in those droplets of water? Bacteria, and a lot of them. So then in the morning, so say you've had rain overnight, or you've had some wind to blow those droplets around, or you irrigated when those guttation droplets were there, the pathogen gets moved around, and then as the plant warms up and the guttation droplets get pulled back in, the bacteria gets pulled back in. So it's landed on a new leaf, it's gotten pulled into these veins, and that's where you're seeing those symptoms. That's why you're seeing those V-shaped lesions from the pathogen getting moved into the leaf. And obviously, the pathogen in here, you're not going to be able to control that in any way with any kind of product. Oops. Here's bacterial leaf spot in peppers, so an example of another bacterial disease where you get these little tiny spots. You get them on the stem, you get them on the fruit. This is another one that's seed-borne. And unfortunately with peppers is you get a few spots on the leaves and then they drop their leaves. So you get a lot of defoliation with bacterial leaf spot. This is septorial leaf spot with parsley. With our different pathogens, they will produce kind of similar symptoms crop to crop. So with the septorias, you tend to get these small gray spots. Sometimes they have a black halo around them, as with the septorial leaf spot on tomatoes, and sometimes it's just a gray spot. But if you look really closely at these, if you had a hand lens, you'd see little black, almost pimply shaped structures there. It's going to take a good hand lens to see those. But if you can see these structures, you've seen the pathogen, and that's when you know for sure what you've got, because that is diagnostic for septoria. Here it is on lettuce. So it's a less defined spot on lettuce, but if you were to look really closely with a microscope or a really good hand lens, in the morning if you collected the leaves when it's been wet, or if you put the leaves in a wet environment overnight, you'd see these tendrils coming out of these spots. So here's a real close-up of those black spots I was just showing you, and you can see these tendrils coming out. What those are is those are spore tendrils. So you can probably imagine as you look at that it looks like it's kind of all, well, they're all glummed together. And they are. So they're not going to get moved easily by wind. But water splash is how they're going to get moved around. So if a splash of water were to come and hit that and bounce off, all those spores are going to go with that water droplet onto another leaf and infect. And a lot of our fungal pathogens can just directly penetrate and get into the leaves. Some of them have to go across the leaf and find the stomates to get in. Our bacteria all need to find stomates or wounds to get into leaves. So another important concept and fact just to be aware of is that our pathogens change. I kind of feel like in my career so far, I've seen more changes than I ever thought I would see in my career. So I started in 1988 at this. We've seen new strains of the late blight pathogen and the cucurbit downy mildew pathogen. They've changed occurrence of these diseases because of these new strains. I've seen new hosts occur, so with Phytophthora Blight that occurs in peppers and cucurbits. Since I started my career, it started to attack beans. And to me, biologically, that's kind of fascinating, but also devastating from a management point of view to realize that, okay, now we got to think about that we can't rotate with snap beans and lima beans because they're also susceptible. And we've got new pathogens in the US. Most notably is this basal downy mildew. And just to show you what that looks like with basal downy mildew, you get this yellow banding on the leaves. Having a banded kind of a look or an angular look to downy mildew is typical, and that's because the pathogen, when it's inside the leaves, it can't move across major veins. So that can kind of help you diagnose a downy mildew disease. Most of our other pathogens like powdery mildew, it's just going to go across the leaf, or late blight on tomatoes, it's going to move right across veins. But you'll get a real angular look with the downy mildews until they've totally consumed the whole leaf. And then if you flip the leaf over, this is why when you're trying to diagnose and figure out what's happening, you always want to see if you can see the pathogen. Can you find it? And often it's going to be on the underside of the leaves. And you flip that leaf over, and it's just covered with the down of the downy mildew pathogen. Now, if you were to look closely at that leaf on a microscope, what you would see is these structures, these kind of tree-like structures with the little spores on them. And so this is a dissecting microscope, and here's a regular compound microscope. So now we're really up close. So you can see this pathogen, it's got like a tree and the spores sitting on it. And that's typical of all your downy mildews and also the late blight pathogen. It's producing these little trees, these little spores on it, and you can probably imagine those are going to be wind dispersed. They're sitting there on those trees, just waiting to get blown away by the wind. This pathogen produces a dark spore. A lot of our downy mildews produce a dark spore. Dark spores are going to help protect them from UV radiation, so they're going to be able to be dispersed longer distances. A really good sunny day will kill them. They won't survive that well, but they can survive a little bit long distance transportation because of those dark spores. So if you can see the pathogen, you can diagnose it, you know what your issues are. There's a lot of spores there. I mean, look at all those spores. There's hundreds and hundreds of thousands of spores, so there's a lot of inoculum there. The other challenge to managing this disease is when you go out and apply a fungicide, you're hitting this side of the leaf. You're not hitting this side of the leaf, which is where the pathogen is. So it makes it really hard to control some of these diseases. This one in particular, I've had a lot of trouble trying to manage it with organic fungicides, unfortunately. So how do we manage it? And this is one I suspect you guys have if you're growing basil. Most of you growing basil. I know there's one grower I know over in the Shenandoah Valley, and he says he's been seeing it every year, late May to early June. So I suspect wherever you are, you're probably seeing it now. So starting out with pathogen-free seed is important, particularly if you're growing in the greenhouse. Because we've got such easily wind-dispersed spores, once we're in the regular growing season, the pathogen's going to show up. Starting off with disease-free plants, this has particularly been an issue with sometimes with plants that are being sold to gardeners. But if you're bringing in transplants, you want to make sure you're inspecting them. We've got resistant varieties coming, some less susceptible varieties. There's Eleonora. It's a little less susceptible, not a lot. And the spice types are definitely less susceptible, but what most people want is sweet basil. There's a breeding program going on at Rutgers. They've got some excellent stuff coming along. It's going to be a few years before we see it. But I've seen, for instance, this last year, some of the material that I looked at from theirs, we couldn't find symptoms at all. I was really amazed, really good resistance. I think a really good option is planting early and harvesting early, and that works really well if what you're trying to do is make pesto, but if what you want is basil to go with tomatoes, that is a little more of a challenge. Be out there scouting. I have a monitoring site so you can keep track of where it's occurring. Fungicides, there's several organic products that are labeled for this. I put question mark because I have not had good control trying to manage this disease. Last summer I had a preventive spray program. We started spraying before basal downy mildew was there. It was a little bit slow to get going for us, so we had, I think it was 5 or 6 applications out before downy mildew showed up. We were spraying twice weekly. I have a sprayer that has drop nozzles, so a nozzle over the top of the plants drops out over the side to try and get good coverage, and we did not get control. I was very disappointed. In the greenhouse, if you're growing basil in the greenhouse and you can reduce humidity with fans and lights, wide plant spacing, drip irrigation, whatever you can do to reduce humidity and keep it below about 85%, you will be golden. Lights on overnight in the greenhouse is a really interesting thing. This comes from some work in Israel, and they've actually shown it's the first half of the night period. What happens is you can take a plant that's infected, and the lights at night messes up the pathogen's ability to produce spores. It needs a dark period to produce spores. And I think that's kind of cool, and really, to me, a really organic way to go after a pathogen, because you're understanding its biology and messing it up. And then at the end, once you realize that you're losing it to downy mildew, you want to promptly destroy that crop, particularly if you've got some successive crops that are coming along. It might help you a little bit. But also because if you're not going to get anything out of that crop and your crop's producing a lot of spores, you want to be nice to the other farmers nearby and get rid of that source of inoculum so you're not messing them up. As I said, we've got a website called Vegetable MD Online. I've got an article up there. It's got a lot of pictures of downy mildew, and I've got links to this reporting site that I've been maintaining. That's a way to know where downy mildew is occurring with other people reporting. And I appreciate if you folks are all seeing it, posting there, or just dropping me an email. It really helps me to understand this pathogen, to know where it's occurring. All right, some other just facts, things that have happened, is to realize that we can get changes in occurrences of pathogens as we get into some different weather, and certainly that's happening now as we're getting into some climate change. So where I am on Long Island, fall of 2003 was incredibly dry, and we had powdery mildew occur on lettuce, which I had never seen before in all of the years I'd been there. I talked to some of my pathology colleagues in the Northeast who've been there even longer than I have. They said they'd never seen it before. This is a disease that occurs routinely out in Arizona, where it's really dry, and that's when they see it, when it's really dry. There's a grower up my way who's now getting into winter growing of greens, and she's been really battling in the greenhouse. So a whole new disease is kind of appearing on us. Not quite sure where it's coming from, if it's coming from a weed. The powdery mildews, like the downy mildews, all have very narrow host ranges. So the powdery mildew on lettuce is different from the one that's on cucurbits. It's different from the powdery mildew that can occur on tomatoes. Some of them have weed hosts, but that's important to realize. Some of our more specialized pathogens, as we call them, these obligates, tend to have very narrow host ranges. The most narrowest that I'm aware of is basal downy mildew. That's all it goes to, is basal. And that's important to know when it comes to management.
Quick question on the
yes.
basal downy mildew management. Will that overwinter? Is that obligate, non-obligate?
It is, God, I didn't put that in the management list. I should have had it in there. It has no way to survive over winter. So our downy, I got to remember to update that on that list. That's a really good point. The downy mildew pathogens, they can reproduce sexually and asexually. So what we're seeing, all those spores, those are all asexual spores that are being produced. In order for these pathogens to reproduce sexually, they have to do basically what we do. They have to be equivalent of a man and a woman coming together. And we call them different mating types. You have to have an A1 and an A2 mating type. With the basal downy mildew, we do not have both mating types in the US. We only have one. The cucurbit downy mildew pathogen is the same way. We don't have both mating types. So they don't have a way to come together and sexually reproduce. And the result of sexual reproduction with these pathogens is a different spore type that can survive without host tissue. It's got a thicker wall. I'll show you, I've got a picture upcoming that shows it with a late blight pathogen. So that's where understanding the biology really helps. Are you working with a pathogen that is able to sexually reproduce and produce a spore that can survive in the soil long term or not? Basal downy mildew, no. But that pathogen moves around. I've had it in my garden every single summer since 2008. And I think any disease that will hit the few plants I have in my home garden tells me there's a ton of inoculum all over the place out there, unfortunately.
You're saying...
Okay, so where is the inoculum?
Where is the inoculum for basal downy mildew? First and foremost can be seed, and then it's going to be wind dispersed spores. So once we're into the growing season, spores are just getting blown all over the place.
So it still means that the original source has to be a infected seed that then infects the plant, and then that disperses by wind?
The original source is going to be seed. So the original source to the US is seed. The original source in a greenhouse during the winter will be seed. Once we're into the growing season, we've got the potential of spores moving the long distance. So right now, basal downy mildew is present in Florida. As we get into the growing season, and it's there every winter. That's where it survives over winter. The cucurbit downy mildew pathogen survives over winter down there. So we get into the growing season, and you're going to start having these pathogens just leaping up the coast. They're producing spores they're continually producing these asexual spores. They land on a new leaf. In a week, you've got a new spot with spores being produced, ready to be wind dispersed elsewhere.
And you're saying with both the basal downy and the cucurbit, they're both reproducing asexually?
Yes.
That's all we have in the state?
That's all we have
And
at this point.
That is technically a weaker form of reproduction because it's not as persistent. It can't survive in the soil without host.
And that's an interesting question. It depends on how you look at it. You could look at it that the asexual reproduction is actually better because the pathogen is just continually producing itself. It's more prolific. And if, say, for instance, you selected on, say you were growing a resistant variety and you had a chance mutation and you end up with a new strain of the pathogen that can infect a resistant variety, and now it just is clonally reproducing itself, you can end up with a big problem. We've seen that particularly with late blight, where you get these new strains come in and whoosh, they just take off. So both forms of reproduction have their value. So the sexual because you can survive over winter, the asexual because you just keep mass producing yourself.
Sexual actually, you're saying, can survive in the soil without host.
Correct. So that's an advantage.
Will not survive in that manner.
Correct.
It's not surviving in debris.
Not basal downy mildew and cucurbit downy mildew are not surviving in debris. Nope.
Now, have you had any success with the fungicides that you had listed a couple of
No.
slides back with
No.
the downy mildew?
No. And the organic growers I've talked to have been pretty disappointed in trying to control it. I think part of the challenge is with something like an herb, one tiny spot in the leaf is as badly infected as a leaf that's completely covered. So when I rate my leaves, I'm looking at percent leaves affected. I'm not looking at severity when I'm assessing it. Because you're not going to be able to market a leaf that's got a little spot. Something like cucumber downy mildew, you're harvesting the cucumbers. So, a few spots on the leaves, that's a different story. Important to keep in mind. Yes?
I was just wondering if heat treatment was not effective on basil seeds.
No, can I get to my one? Heat treatment. Heat treatment is effective. The problem with basil is when you put it in water, it exudes a gel-ish substance, a gelatinous substance, which makes it hard to plant. There is a steam treatment. ENZA has developed a seed treatment that's steam. So there's an option. If you're getting seed from ENZA, they will treat the seed.
What's the name of it?
The seed company is ENZA. ENZA does a steam treatment of their seed. They're the ones who produced Eleonora.
Would you spell that, please?
ENZA, E-N-Z-A. Okay, so the other thing to realize, I've kind of gone over some of this with some of the other slides, is that our pathogens have different host ranges. They go from narrow to broad. The cucumber downy mildew one is interesting because it exists as pathotypes that infect different cucurbit types. All of these pathotypes affect cucumber. Some of them also affect summer squash or butternut squash or melons. And that's why you will see cucumber downy mildew always on cucumbers, sometimes on the other crop types, sometimes not. The cucurbit powdery mildew pathogen also infects the ornamental verbena. So here's one that really makes me scratch my head. I mean, why does this pathogen infect such totally different plants? But it does. Phytophthora Blight. As I mentioned before, it goes to cucurbits, peppers, eggplants, tomatoes, and now it also goes to snap bean and lima beans and some weeds like purslane. That's a really diverse host range. It doesn't go to potato, another solanaceous. Another one, scratch your head about. And then white mold goes to hundreds of different crops and weeds, which makes it harder to manage. So, what's our rotation? Really important management practice, particularly in a biological system. What are our challenges? Host range of the pathogen. That's going to dictate what you're going to rotate. You know, our usual thought is to rotate amongst families. Well, as I just showed you, sometimes that just is not going to work with a pathogen. If a pathogen can survive really well in a soil for years, such as the white mold pathogen producing those sclerotia, it's going to be really hard to be successful with rotation. And then dispersal mechanisms. If you've got a pathogen that can be dispersed by air, then if you manage it on seed, it's going to, you're going to have it come back in again. So something like basal downy mildew, you start with clean seed during the growing season, you're going to have wind dispersed spores coming in later on. So clean seed gets you off to a good start, but it's not the only solution to managing that pathogen. And likewise, if you like hot water seed treatment for tomatoes, really great going after something like your bacterial diseases and septoria, but if you don't rotate, then you're not going to control it. Or if you're rotating well, but you don't clean your tomato stakes, some of these pathogens, particularly the bacterial ones, can just hang out and live and survive on your tomato stakes. So if you haven't disinfected them, but you did all the other steps, you rotated well, you cleaned the seed, you're still going to have a problem. So you need to understand all aspects, all ways the pathogen has to survive and be dispersed. And then some of our pathogens just are able to explode, which I think makes them really hard to manage with rotation. And a real prime example of that is the Phytophthora Blight affecting the cucurbits and peppers. I've seen that one just produce spores and just really take off in a field. Realizing that plant diseases can't be cured. Fungicides, especially our organic ones, they're going to act on pathogens prior to infection. So it's really going to be critical to success of a fungicide program that you're starting really early in disease development, and preventive if you can. The other thing to realize is that there is a lag time between infection and when you see symptoms. Biologically, we call that the latent period. So therefore, it's really critical to start your fungicide applications early in disease development, preferably before you've seen symptoms. And here I'll give you an example. Here's downy mildew on cucumber. Here's first symptoms. These symptoms appeared about 6 days after spore dispersal to the area. This is a cucumber plant in my home garden, so it gave me a chance to kind of monitor it and see what was happening. I was not spraying anything for it. This is what downy mildew looks like. So here are some of those first symptoms, what you'd be seeing. So you get these little yellow spots. Now if you look up close, a lot of them are stopping at the veins, so they're very angular looking spot. As I said before with downy mildew, initially it does not get by those major veins, so you get that very angular look. And then if you look at the underside of the leaves, you'll see this sporulation down there of the pathogen. So just like what I was showing you before with basal downy mildew. There it is on cucumber. And here's some really early symptoms. They might not quite look angular. They're just reaching that point. Those are the kind of symptoms you really want to know and recognize. There's one that's a little more characteristic. So there's that plant, and here's the plant about a week later, and look at how many more symptoms are here. Those infections had already occurred back here. So you're spraying to try and control downy mildew here. That's really the amount of infections you've already got that you're not going to touch with an organic fungicide. Really important to keep in mind. And just to show you how fast the disease can go, that's just 4 days later. Some of these diseases can just be tremendous in how fast they move. So I think if you're going to use fungicides, you've got to have really good timing. So I think now's the time to decide, what diseases do you want to treat with fungicides next year? What do you feel is having enough of an impact for you that you need to try to stop with fungicides? Decide now what products you're going to use, where you're going to get them from, so you don't have to think about this during the growing season. You've already decided, for instance, okay, I'm going to use Actinovate, and this supplier has some, that's where I'm going to go get it. Early in the season, get your sprayer ready, get the nozzles, get it calibrated, replace old nozzles. The year that we had our big late blight outbreak, 2009, up in the Northeast, a number of growers weren't ready to spray because it was June when that disease came in. Gosh, we're just barely getting done with planting tomatoes at that point in time. So a number of our growers, particularly our organic growers, weren't ready to spray. They had to get their sprayer ready, they had to get products in, and by the time those couple of days went by, they were way behind it and just could not save their tomato crops. And that was a sad thing to see. You want to be scouting routinely. We do have some disease forecasting programs that can help you know when diseases are present, when it's time to spray, and monitoring occurrences. We've got some websites that will help you with that. The disease forecasters, what they're doing is they're going to help you time fungicides applications when they're needed most. They're based upon whether the pathogen is present with some of them, and that's the cucurbit downy mildew one, is the one that's considering where the pathogen is. They're looking at what conditions are favorable for the pathogen, and they're looking, some of them, at how the fungicide weathers, and that's the case with the late blight one. So that one will consider if you've gotten 2 inches of rain, that's enough to take off the fungicide residue that's on your plant. That will trigger it to tell you, oh, you need to spray again, you've lost all your residue now. And favorability of conditions, they're looking at favorability on your plants, most of them. Do you have the right kind of conditions, the right moisture on the leaves, the right temperature for the pathogen develop. With the cucurbit downy mildew one, it's also looking at, are the conditions favorable for spore production where we know the disease is occurring? Are conditions favorable, it's looking at where the wind goes. Are conditions favorable for the pathogen to survive wind dispersal, which would be having nice cloud cover to help protect those spores from UV radiation? So that one's taking in all those factors, which is pretty interesting and neat. Downy mildew can be pretty devastating. So here's downy mildew on acorn squash. If you lose your leaves on acorn squash really early before it's had a good, I think it's 60 days it needs, if I'm correct about that, from when the fruit is formed until you've got your high levels of sugars. You got to have enough time period to get the sugars accumulated in the fruit. And if you haven't had that time period with good green leaf tissue to be providing the photosynthate, your squash is going to have absolutely no flavor. So important to control, disease to control. This disease is a really excellent one for a forecasting system because it's got no way to survive over winter anywhere that the plant can't survive, so that would include here, because it can't sexually reproduce. It's got no other hosts, and it's not seed-borne. So unlike the basal downy mildew, this pathogen hasn't figured out how to get into the seed. So its only sources north of Florida, where the crop is growing now, is going to be long-distance wind dispersal of spores, those asexual spores. Infected transplants is really rare, but we had 1 year up in the north where some growers brought in Florida-raised transplants, and they brought downy mildew in on them. And this forecasting system is unique because, as I was just describing, it's predicting pathogen movement as well as conditions favorable for infection. So it's pretty neat. So the pathogen can only survive below the 30th latitude over winter. And it really varies when it can show up here in the north every year. So this is when I've seen it on Long Island the past few years. And notice I've seen it every single year. It's going to show up eventually. The earliest I've seen it is July 17th, and the latest I've first seen it is September 7th. That's a pretty wide spread. So when you've got a disease like this that's going to show up over such a wide range of time periods, I think it's really helpful to be looking at a forecasting program to know when it's going to show up. And there is such a thing. Here's the cucumber downy mildew forecasting page. So you can sign up to get alerts there. You go to that one spot and click, sign up for alerts. You put in where you are. Tell me where the disease is relative to where your farm is. What do you want to know in your alert? Do you want to get it by email, by text? You can sign up for all this. Doesn't cost you anything. This is what the maps look like. So you can also just go to the web page on a regular basis and see, and it's going to tell you where disease is occurring, what the risk level is from low to high, and it also describes what's happening. So the other kind of information that's there that's helpful to look at is your epidemic history. So you go to the page, you can click and see a little, well, here it is, show you up close. You can see if you click to look at the history up close, click on that button, you're going to get all this kind of information on a daily basis, new reports coming in. So let's look a little closer. First reports are in red. New reports will be in blue, telling you of a new report in a state. And then each of the hosts is color-coded as to what it's being seen on. And that's important because, as I mentioned before, this pathogen exists as different pathotypes. Cucumber is infected by all of them, but depending on what pathotype is present will dictate what other cucurbit types are going to be infected. So when you're looking at that forecasting program, you want to know not only are your crops at risk that the pathogen might be coming, but what crops has it already been seen on? And so therefore, what crops of yours are most at risk? If you're only seeing cucumbers in that epidemic history, then you're only concerned if you have cucumbers. Yes?
And that program is built by all extension services on the East Coast?
That program is run by North Carolina State University. They run the program, but it's pathologists such as myself who log reports anytime we see it. The other way we support this program is a number of us grow what we call sentinel plots, where we grow all these different cucurbit types so that we can be monitoring what pathogen types are out there to get that
in
info.
Virginia, we would need our extension to have people doing what you're doing.
And you do.
Okay.
You do. All of this.
Either at BSU or across the state.
I don't remember who is got I think there's a couple sentinel stations here in Central Park and also over on the Eastern Shore. I know I've seen reports listed there from various places. But what really helps is also for growers to be putting in reports. So if you see the disease on your crops, think about how much you benefit from other growers reporting. So, it's part of that community. If we all help and all get in reports, that will help a lot. This is just an example from several years back, 2005, where we had a situation where we had some tropical storms swirling around near me in the mid-Atlantic region. So based upon the environmental conditions we had, I thought for sure we'd get hit with downy mildew on Long Island, so I was nervous. If I did not have this forecasting system, I would have been telling my growers, you need to spray. We know we've got downy mildew south of us. We've got these conditions. Certainly we've got risk. But as I looked at the risk description, there's no risk mentioned here for Long Island at all. And looking at what the wind trajectory was, now back then they were showing it differently than they are now. They were showing a line. But you can see they were projecting that this is where the wind was going to go from the known source in South New Jersey. Not going anywhere near us. Here we are on Long Island. So that's why we had no risk. The wind trajectory was not coming our way. So that's where this forecast is really helpful. Well, a little later in the summer, that's where the trajectory went. That just happens to be where my home garden is, and that's what happened to the plants. So forecast worked. Now, we've got an organic grower upstate New York, Lou Lego, who has another interesting approach to take to managing downy mildew. Most people will take those forecasts and say, okay, now it's time to spray for downy mildew. What he did, because his background is in engineering, is he developed what he calls a spore exclusion tunnel. And what he did is put these HEPA filters and set up his tunnel so that he could turn it on so that there would be negative pressure, which would force the air only to go through these filters to get inside of the greenhouse. His protocol for how to set this up is all available from him. If you're interested in trying it, he had total and complete control inside there. So he would monitor, watch the forecast. If the forecast was high, he would turn on the tunnel so that it was under positive pressure. I think it's positive, negative pressure. I don't know. I'm not a good engineer, but whatever it takes to force the air to go through these filters and close up the greenhouse. He had plants outdoors. They became infected. He had plants underneath high tunnels or low tunnels, the floating row covers outside without this, and he had downy mildew in those. So it was completely this ability to exclude the pathogens from getting inside. And that ought to work for basil downy mildew if you wanted to grow basil in an exclusion tunnel like this, or late blight in tomatoes, any kind of spores, wind dispersed spores that should work with. So other ways to manage downy mildew is resistant varieties. At this point, resistance has only been bred into cucumbers. The fungicides is a number of different products, the forecaster. Remember, good coverage is always going to be critical. And then planting early, since the Downy Mildew pathogen is going to get wind dispersed from Florida, it's going to take a little bit of time. Focusing your production earlier in the season helps. Not so much when you're trying to provide fruit, particularly cucumbers, through CSA or roadside market, because consumers seem to always want cucumbers. There is resistance that's been bred out there. A number of varieties have resistance to, these guys right here, were developed for resistance to the old strain of the pathogen that was here back before 2004. They're still showing some resistance. This is a line that's coming out of the Cornell plant breeding program. That was most resistant, one of the most resistant ones in my trial last year. And then there's 2 numbered ones that are coming out of a seed company, and they did not look so good last year. This is what the fruit looked like. In the larger ones that are in larger print are the ones that did the best for me. I really like Ivory Queen if you got a market for the white cucumbers that had really good resistance. This line from Cornell was really holding up well at the end of the season. It's a little bit later. Hopefully we'll see some varieties coming out commercially of that one in the near future. Just to show you some data from some trial that I did a couple of years back, looking at an organic fungicide program applied to varieties that have resistance or don't. And this is just to show you that sometimes an integrated management program, particularly an organic system, can really be the way to go. So what I was using is a botanical oil organocide with a low rate of copper on a variety that has absolutely no resistance, this straight 8. Severity was 52%. That doesn't sound like very good control. So 52% of the leaf tissue had symptoms. But these varieties that have old resistance in them, so that don't stand up by themselves to cucumber downy mildew, sprayed with an organic fungicide program, there's less than 10% severity on all of them. So an integrated program, you can get really decent control of downy mildew on cucumbers. And then just to show you some data looking at different products, different organic products. This is just to show you that there's some real variation in how well some of these products work. So for instance, in this trial, I had Serenade Max. These are the statistically significant letters here. Now we're looking at percent control. So the smaller the number, the worse. The bigger the number, the better control. So with copper, I'm getting 50% control. So A means ineffective. So for me, Serenade Max was ineffective. You can see I range from Actinovate giving me 26 control to Spore-Tec giving me 61% control. So definitely variation in how well these products work. I want to shift gears and talk a little bit about late blight in tomatoes, which has been a really big disease problem for us in recent years on Long Island and in the Northeast in particular. This is a really destructive disease, and it's a really contagious disease. And this is the one that we all in the Northeast particularly consider a real community disease, and talk about that we all need to manage it together. We need to be communicating amongst each other so we know when the disease is occurring and we can alert each other. And when, as a grower, early in the season, you've got an uncontrollable outbreak, you act and get rid of it. That can make a huge difference. The late blight pathogen has been traditionally more of a potato pathogen. It's been more of a potato disease. And for a couple of reasons. Number one is that this is an obligate pathogen, so it needs living host tissue. So those leaves, when they die at the end of the season, so goeth the pathogen. But what's living tissue of a potato over winter? Potato tubers. So that's how that pathogen survives over winter. Either in seed that you purchase, seed that you save, potatoes that don't get harvested, stay in the ground, potatoes in a coal pile. All ways that the pathogen can survive. So those are our primary sources. There's an infected potato tuber, sprouts coming up, so the pathogen's in the potato seed piece, it grows up the sprouts, and then it's going to produce its spores on those leaves. Those are what the spores look like. Wind dispersed spores from plants in frost-free areas is your next source. There's the spores. You can see they're white. They're a little less tolerant of being dispersed long distances. They're not going to move as easily as our downy mildew pathogens. They'll move maybe 30 miles in a jump, generally not more than that. Infected tomato transplants, we've definitely had a couple of years where that's been our primary, particularly being sold to home gardeners. We had 1 year where we had a strain of the pathogen that was able to infect petunias. So that's where you get these strains that can go to less common hosts. So that was a source 1 year. I'm going to go over I figured it was good to go over one disease cycle to show you what these disease cycles look like, and late blight pathogens are a really good one to do with that. You will find these on the web. This is your way to really look at the biology and understand the biology of these pathogens. So here's the late blight one. So as I said, it's going to survive in the potatoes. It grows up in the spring up onto these sprouts. And then it's doing this asexual reproduction where it's producing the spores, they're when dispersed to a new leaf, and this continual cycling. And then come the fall, the leaves that are infected, particularly once it's on the stems, the spores can get washed down to where the potato tubers are and infect those potatoes. And then you get the cycle all over again. So that's the late blight disease cycle. There's a leaf infected. There's a lot of spores there. There's up to 300,000 spores there. So that's a lot of inoculum potential. That's where you really get this explosive disease potential. And with this pathogen, depending on the conditions and the pathogen, it can take 4 to 10 days to complete the cycle. So it can be as little as 4 days from a spore being blown from here, landing on a new leaf, and producing a new spot that's got more spores. Pretty short. Now, the other cycle that can occur is that sexual cycle. We do not believe that this cycle is occurring in the US at this point in time. So that's the sexual reproduction cycle. If it did occur, you can see what happens is you get this thick-walled oospore that forms, and that spore can survive in the soil. As I said, we do not think that this happens at this point in time in the US. It does happen in Mexico, some South America, Latin American countries. In recent years, it started to happen in Europe because they brought in some potatoes with the other mating type. So they got the two mating types together, and now they have sexual reproduction occurring there. They see late blight a lot more often. The other thing that we don't like about having a pathogen sexually reproduce is that you're going to get more variation that's happening. And then what that can mean is you end up with more genotypes. So this pathogen, so genotypes, what are genotypes? Genotypes is the strain, so it's the genetic makeup of the organism. The late blight pathogen tends to exist as a real clonal population where there's just no variation. We get powdery mildews, and there tends to be a little bit more variation in the population. So your squash leaf that has powdery mildew, it's going to look a little bit more like this room with some diversity, but a lot of duplicates of each of the different powdery mildews that are there. But if you were to look at that leaf, if you look at your field that's got tomatoes, every single leaf, every single pathogen that's out there would be absolutely identical. It's all the same genotype pretty commonly. So at this point in time in the US, we've had 24 genotypes described. That's not very many in a really long history of having this pathogen present. In most years, we have 1 to 4 that dominate. So it's a real clonal population that's occurring. And knowing the genotype tells us a lot about the pathogen. Its preferred host, ability to overcome resistance. So where do we get new genotypes? You can get a chance mutation during asexual reproduction. And sometimes that's a mutation that is beneficial to the pathogen, a lot of times not. And then you can get variation during sexual reproduction, and that's really where you're going to get a lot of variation. Just think of you and your kids, or look at your parents, look at how much variation. Same thing with the pathogen. When it sexually reproduces, you're going to get a lot of variation. So, strains that have occurred recently in the US, the US 8 is the one that's been around for quite a while, and then we've gotten these others that have dominated the past few years. This one, the US 8, really liked potatoes. And that's one of the major reasons, the other major reason why late blight has not been a big issue on tomatoes until recently. This strain really didn't like potatoes. 2013, similar last year, only 2 of the isolates that were tested were found to be the US8, so it's really disappeared. This one was really giving our conventional growers a lot of headache because it was resistant to a fungicide that they were using. And that's why it got selected, because of their use of that fungicide. This, having strains out there that are sensitive to this fungicide, why it doesn't matter to us as organic producers, it's valuable to know when there's resistance issues for conventional growers, because if they're having problems and they can't control the pathogen, then there's more inoculum out there that's going to end up on organic farms. US 22 is the one that caused all the headaches in 2009. It's pretty much disappeared. It really liked tomatoes. The one that's dominated recently is this US 23. It likes both potatoes and tomatoes, which is why we're seeing it quite a bit. It's a different mating type. This makes us nervous. So we've got different mating types in the US. Generally speaking, we almost never find them in the same field, thank goodness, but that potential does exist. Most of the isolates we're finding are this US 23. So that also helps bring down the possibility that we're going to end up with sexual reproduction in the US. This is another reason why it's really important to let Extension know when you've got late blight, so they can get samples, they can get them tested, find out what strain is present. And if they start to realize that there's multiple genotypes of different mating types in your area, then that's when we really want to get on and be aggressive about managing this disease, even more so than otherwise. This graph just shows you differences amongst these strains over time, looking at potato versus tomato. You can just see the variation in what strains are occurring. They definitely, some of them prefer potato over tomato. And then the other thing is just to look at how things change over time. So this US 23 appeared in a little bit in 2009, but you can just see how it's exploded. And in recent years, it's our dominant strain. So that just shows you how this pathogen can change. You know, the populations can change over time, and that will affect whether or not it's occurring on these particular hosts. So just to show you, this is a chart with when we've seen late blight first on Long Island. This really documents how late blight has changed in occurrence over the years. So from 1996, 2008, you know, there's a lot of years in there where I didn't see late blight at all. 2 years where I didn't see it until October. At that point, it's pretty late in the season. It really doesn't matter. Since 2009, we have seen it every single year, and we always see it pretty early. So the pathogen has really changed in a lot of places. Some years we've known for sure the source has been gardens. These 3 years, the last 3 years, I have no idea. I've tried real hard to figure out what our source was. I don't know what the source is. This makes it hard to manage it when we're not sure if it's starting in volunteer potatoes. Is it continually starting in gardens? Where is it coming from? You can get information about occurrence of late blight each year from the usablight.org webpage. And this definitely shows you that it has been really a New England, Northeast rather, problem. You can go here and click on this group by state to get everything grouped by state, and then click on your state. So I did this for Virginia for 2014, and you'll see that there were 4 samples that were submitted, probably a number of other cases that occurred. So you could go there and see during the course of the season where late blight's being reported so far. So these reports started in July, end of July, and the 2 that were tested were both the US 23. If you go to the website right now, there it is in Florida. So it's brewing in Florida. It's present, so time to start monitoring. When it comes to management, I talked a little bit right at the very start about what we call the disease triangle, those things you need in order for disease to occur. You know, having the pathogen, having the host, having the favorable environment. You got to have all 3 to have disease, plus time for the disease to develop. So when it comes to management, you know, we try to control all 3 of these. So just as an example, with tomatoes and for late blight, having the plant staked is going to help. Having them up, having good air movement will help a little bit. It's not going to be a panacea for late blight, that's for sure. So another good approach would be trying to reduce the susceptibility of the host. And a good way to do that is through having resistant varieties. And we've got major resistance genes that have been identified for resistance in tomatoes. Typically where plant breeders find resistance genes is they turn to wild relatives of the crop. And where do you find wild relatives? You find it where the crop originated, and that's often where the pathogen also originated. So the pathogen and the host have been in this little genetic war for years, and they've developed resistance genes, and the pathogen has developed virulent genes in order to be able to infect. So what the host is doing is it's trying to always be able to detect that pathogen when it infects, and the pathogen keeps changing itself so the host can't recognize it. And you get into this little war, and it tends to be a real gene for gene between what the pathogen has and what the host has to be able to detect each other or to get by the detection. So the genes that are out there all came from this wild relative. It is a different species. The breeding work that's being done now is all through conventional breeding. There is efforts underway. In fact, it started in Europe. There's some people in the US who are also playing around with trying to do this from genetic engineering perspective. I just put that out there so you're aware of some things that are happening. And what they're doing is they're looking at the fact that we know the genome of the potato and the tomato and the genome of the pathogen. So now they can start detecting these genes and figuring out what does the pathogen need to infect? And they're starting to understand all these different genes that are involved in this genetic little battle that goes on so that the pathogen can be a pathogen. Because if you think about it, the disease state is an unusual one in that, you know, this pathogen, it can't attack cucurbits and it can't attack beans. You know, there's a lot of other plants that are just non-hosts that immediately recognize it and prevent the infection. So what some of these molecular biologists are trying to look for is, you know, what is it that makes these non-hosts non-hosts? You know, how are they recognizing it? What does this pathogen absolutely need to infect? And can we tailor the tomato and potato so that it recognizes every single Phytophthora and no infection can occur? But that's another story. What you'll have with plant breeding is your resistant variety will either be botanically what we call heterozygous or homozygous. And that means it either has one copy of the resistance genes or two copies. And that can be important to be aware of because there's a feeling that if you've got two copies, if both parents, when they cross these tomatoes, had the resistance gene, that you should have a higher level of resistance. It's a tougher thing for a plant breeder to do, to bring those two genes together, however. Here's what's out there. So these are what I looked at. I've been doing some variety trials for a couple of years. So there's plants out there that have the PH1, PH2, the PH3, or both genes in them, and then some things that people have reported to have resistance. So I wanted to look at all those. I've been doing this work in our organic research block where I am because it was my feeling that organic growers would be most interested in these results since they don't have the conventional fungicides. There's a whole slew of conventional fungicides that are now available that can get inside of leaves and conventional growers can get a little bit better control of late blight. So I'm going to run through some of the varieties that I looked at, and Mountain Fresh Plus was what I used as my control. So here's New Yorker, no difference. PH1 gene is not effective against the current strain of the pathogen, clearly. Here's Legend with the PH2 gene. There is some late blight there. Let's get a little bit of control. Here's the PH3 gene, which is in Plum Regal. Plum Regal has two copies of it, so this is homozygous. There's a little bit of late blight there, but certainly much better control. The other thing I'll point out is you're seeing some green tissue up here on the top. I did start an organic spray program as late blight started to really take off, and I got a little bit nervous about having impact on other growers in my area, so I thought maybe I need to slow this down a little bit. So I think we got a little bit of control on the top of the leaves. Here's Defiant, which has both the PH2 and 3 genes. Really good control. I've been impressed with the control you get with both of these genes. Juliet is reported resistant. Clearly it is not resistant. And then you get varieties like Jasper that we have no idea what the resistance is, but it clearly does have resistance in it. This is a variety from Johnny's, cherry variety. And then here's a couple pictures from 2013 when I looked at Iron Lady. This is got 2 copies of the PH2 and the PH3 genes, so this is a homozygous one. In my trial, I did not see better control, and it may be because I'm seeing such good control with just 1 copy of the PH2 and the PH3 gene. This variety I don't recommend. It's a new early variety in the breeding program, and the fruit quality is not quite there, but we're working on it. Takes a lot to get good fruit quality and resistance. Mountain Magic is a really nice variety. That is the Campari type. That's done really well in fruit evaluations. This is one of my absolute favorite varieties. Lemon Drop is the largest size yellow. That's done really well. I like that one a lot. Had no idea what its resistance is. If there's any varieties that you've grown, some of these heirloom types that you think has some resistance, I am always looking for new things to test. I have funding again this summer to evaluate varieties, so please tell me if you got something you think is resistant and I'll take a look at it. Jasper looked great in 2013. This is a really nice variety. I really like this variety. So one of the organic growers on Long Island grows a lot of different varieties. He had 60 varieties last summer. He grew every single one of the late blight resistant varieties, part of his insurance to ensure he had some tomatoes. The most growing of resistant varieties that I'm seeing is our organic growers. They're doing it kind of for insurance. It may be their last planting of the season, but they're doing it to make sure they've got tomatoes. When he did his taste variety trial with his CSA members, he had 40 varieties at that point. Jasper was number 1, voted number 1. It beat Sungold, which has been the reigning king, which really surprised me. But Jasper is really good. It holds its flavor late in the season. It's a nice cherry. It also doesn't crack much. So these are some tomatoes in a harvest bucket from my home garden. You can see a crack or 2. I cluster harvested those. So they'll hold on the vine and you can cluster harvest them, which is really nice. Here's some other varieties that I've looked at that look pretty good. Some from Tom Wagner's. I need to do a little more evaluation. That was just an observational last year, but I think there's some other resistance out there. So that's why I'm really eager to look at anything else others of you have seen. None of these varieties are immune. You're never going to see immunity with resistance. You're always going to get some level of disease, which is where an integrated program is going to help if you have no tolerance for disease at all. So what I would recommend is nothing that has just the PH1 or PH2 gene, PH2 and PH3. This is your only plum that's out there right now, and then some of these others. So what else are you going to do for resistance management? If you are growing potatoes, you want to make sure you've got certified seed. Less likely to have late blight in it. Don't save your own if you had late blight in your field. Some potato varieties have what's called a rate-reducing resistance. They won't give you a lot of resistance, but they'll give you something. Make sure in the spring you get rid of cull piles, and if you see any volunteers, get rid of them right away. Don't wait to see late blight appear on them. Some weeds are susceptible, so there's late blight symptoms on bittersweet nightshade. When you're out scouting, you want to look, and this is going to be true for a lot of diseases that need a little bit of free moisture on the leaf or high humidity to infect. Any areas that are shady, if you've got a spray program, an area where the spray boom might not have overlapped is a good area to look. The other thing I found with late blight is sometimes it will start in a high part of the field, and that might reflect how wind has gone over the field and maybe it's clipped. Like I've seen it start at a dome high center, and I've seen it, such as in our research station one year, the first field it picked up was at the end of our farm where the land starts sloping up, and I think plants were at a point where they kind of caught the spores. So high areas can also be a really important place to look. It can really help if you find it in a hot spot to rogue out those plants and destroy them. Late blight is a community disease. If you find it, make sure you let Extension know that you think you've got late blight, and make sure you let the farmers nearby know so that they can be aggressive about managing it in their plants. Newsletters. Every state's got an Extension newsletter. They can, in my area, if I see late blight, that is one disease where I will put out an alert. I won't even wait till I get to Thursday when it's time to write my weekly newsletter. The information will be out there. And the usablight.org website. You can sign up to get alerts there and know every time the disease is posted near you. Whoops. So minimizing leaf wetness will help a little bit, but this pathogen just needs high moisture. And actually what you can end up with is if you don't have plants on plastic, you can end up with a higher moisture situation than when they are on plastic. So you can get more disease issues in a situation like that. Applying fungicides on a preventive schedule is considered essential with late blight, just because way too often when it first appears in a crop, there's too many symptoms to stop it. And I've seen that situation happen too often. Getting good coverage. If you're a really small scale grower and you're hand spraying, you could do what I do with my research plots. I have a boom that's got just a single nozzle on it that's got a wide, and it's a twin jet, so it's spraying like this. And I hold the boom sideways. So I'm going along and I'm spraying along the side of the plants to get down, to get that whole canopy. Because a lot of these diseases, where it's going to be most humid is down here, and that's where they might start. So spraying over the top of the plants, you're not getting the coverage you can get if you get to the side. The forecasting system, there's a forecasting system for late blight, and then destroying the crop if it gets too severe, just so you're not having an effect on others. There's been some efficacy work done on various products. Coppers tended to be really the best. I've seen good results with actinovate. The EF400, oxidate, I've seen mixed. Regalia, Zonix, one study, and that one didn't work in that study. So just to give you an example, untreated, the amount of disease, copper, the amount of disease, oxidate. So quite often, coppers tend to be the best. And here's a study with actinovate, also out of Florida, getting some control. There is a decision support system that will help you know when to spray. And what happens with these decision support systems is they're going to tell you when you need to spray less often and sometimes more often than weekly, depending upon the conditions. So hopefully the net result is better control and fewer applications. I think there's a real challenge when you're in a diversified system. I know with the growers I've worked with on Long Island, they tend to have one really good day for doing their spraying, and it's hard to break away from that. And it may be that you want to spray the day before you don't have workers in the field. It's when a lot of my organic growers in particular are spraying, which makes it tough to do this. But late blight can be a really good one for decision support system because it is hard to predict when it's going to start. And it is really hard to try to manage it with fungicides after it's started. And it can be hard to tell when conditions are favorable, when you got high humidity. And I've seen that particularly in August. We'll go days and days with no rain, so you kind of go, it's not disease weather, but we're still getting high humidity, especially at night when we're all sleeping, not paying attention to the humidity, and that's when our pathogens are infecting. So you can get to the Cornell's decision support system here at the USA Blight website. You plug in various factors when you're applying your fungicides, when you irrigate, you can do it on a smartphone. The other thing they're putting in now is an infection alert. So they're starting to look at and pull in ideal conditions for the late blight pathogen and where it might spread across fields. But you can also kind of look at what we know biologically and know that, okay, on a sunny morning, you're going to have more spores released. But a really sunny day is not going to be good for the pathogen to move out of your field and really do a lot of moving, or move from like a neighbor's field that has late blight to your field. Gets cloudy midday, pathogens really survive well. Wet leaves at night or really high humidity, spores are going to germinate and infect. So those are your ideal conditions for late blight. We got some diagnosis challenges with any disease. I'm going to just go over a few here with late blight. You got your typical symptoms. We get these large spots, kind of a wilted edge to them. The spores on the back side of the leaf. Here's another example, really large spots, wilted tissue, spores on the back side. But you can also get very early on just these wilted little tiny spots. Those are very early late blight symptoms. Things to be aware of. And I have all these pictures posted up on the web if you want to go back and look at them later. That sure doesn't look like late blight. That just looks like a lot of dead tissue. I scratched my head. I had to look at these leaves under a microscope, and I was able to find the pathogen on the backside of every single one of them. So that's late blight. Late blight's most commonly confused with drought stress, but you'll see all the symptoms tend to come in from the edge of the leaves. You got a yellow border, not the wilted border, and you're not going to see the spores. And the other odd symptom I saw a couple of years back was this ring spotting, which is a lot more like you see with some virus diseases. That's a really unusual late blight. Notice the size of the late blight spots. These are all Septoria spots, much smaller, never as big. There's my photo gallery where I've got these pictures posted. Go quickly through some of the biological fungicides that we have out there. There's a lot of products now. I've got lists that I'm in the process of putting up at my website. So anything in green you can apply just is labeled for leaf tissue. Brown is a soil product, and blue can be applied to both. So we've got a lot of products that have the bacteria Bacillus in them. Number of other type products. Here's Contans. That's that one that's really good for white mold. We've got one that's good for nematodes, a number of different products out there. So those are our biological-based ones. There are also a lot of organic biochemical fungicides, which have a naturally occurring substance as the active ingredient. So some botanical oils that are out there and available. Most of these are exempt from EPA registration. Some more types of materials, so potassium bicarbonate, your hydrogen dioxides. So a lot of different products now available for you. So those are what we call our biopesticides, and your other organic products that are available. There's several coppers, sulfur, here's a mineral oil, and a petroleum oil. Organic coppers have been kind of the main product that organic growers have used. We've got the most efficacy data with them. They've got the broadest spectrum of activity. So, and they differ in what type of active ingredient they have. They're not all copper hydroxides. So certainly, one of your goals as an organic grower is to minimize how much copper you're putting out there, how much copper you're getting into the soil. So you might want to look at the active ingredient, what percentage is there, and you say, whoa, 98%, that's got to be the most. But what you really need to look at is the metallic copper equivalent. So now this one's the most, Nordox, and some of these are pretty darn low. Oh, come on, thing's getting tired. Uh-oh. Did I kill it? We'll use the computer. All right, so I forgot I had these labeled here. So there's your Cueva's got the least, Nordox has got the most if you're looking at metallic copper equivalent. This is where you're going to find the information on the label. It will be right under the active ingredient. But actually what you need to think about is how much copper is in the spray you're doing. So if you're just looking at metallic copper equivalent, you'd say Cueva's the least, got the least, that's the best product to use. But if you look here and you're looking at your maximum labeled rate for a couple of different crops, so this is the labeled rates, the highest labeled rate, and then in parentheses is your metallic copper equivalent. So basic copper 53 is actually the highest amount of metallic copper equivalent. And your lowest is this one, Camelot. So you can see these are, Cueva comes in pretty good, but it's a little bit more than that one. And then Badge is pretty good, at least on broccoli. And I've got an article up on my website that talks about this a little bit more. The other thing to be aware of is that these products differ in their re-entry interval. So they range from 4 hours to 48 hours. We got a lot of organic fungicides that are available for cucurbit powdery mildew. Biological ones, these are some of those chemically based, naturally chemically based ones. Sporotech I have in red because that's kind of dropped off the market, a little hard to get a hold of now. What's looked really the best, absolute best control is sulfur. That does a really good job, and that's able to get to the underside of leaves. Some of these products are also labeled for cucurbit downy mildew. One that is different, but you'll see a lot of the products that were here for cucurbit powdery mildew are not labeled for, or don't have activity for cucurbit downy mildew. And then there's one product that's only labeled for cucurbit downy mildew. Another management practice for powdery mildew is resistant varieties. There are some really good resistant varieties with the melons. They've done really well in my variety trials, a lot of different types, particularly personal size. That's pretty good, a real good choice for a CSA box. Your resistant varieties, just to understand them a little bit better. With melons, it's a race-specific resistance. We understand the race that's gone on between the pathogen and the host in developing these different genes. So when that happens, we then describe the pathogen as races, whether it's able to overcome different resistant varieties. Races 1 and 2 have dominated, so if you've got a variety that's got resistance to races 1 and 2, it's going to do really well. And if we get back to the slide where I had these, most of these varieties have resistance to races 1 and 2. With the squashes and pumpkins, there's one major gene. We don't have that race description. The best ones are the ones that have homozygous resistance. They don't have the level of resistance you're going to see with melons, unfortunately. Not quite as good. So I've seen variable performance and not as good recently. Just if we got a minute or two for a couple last.
Give you 5. How about that?
Okay. I always have too much to say when I go through all these slides. Realize some copper fungicides can be applied with biological fungicides, either because the organism is tolerant or the active ingredient in the biological fungicide is actually chemicals produced by the biological agent itself. And the major example is serenade and sonata. It's not those organisms that are doing the work. It's what they're producing in fermentation. Keep records. Know when you're first seeing the disease, how severe it gets, so you know, do you need to control this disease with fungicides? And if you are using fungicides, what kind of control are you getting? The other thing to be aware of is that your fungicides approved for organic production are not devoid of potential harm to you or the environment. Realize that copper can cause irreversible eye damage if you put it directly into your eyes. So read the label, know what you need for protective stuff. Realize the label's a legal document. You'll have first aid stuff there. Personal protective equipment, what you need to wear when you're applying the chemicals. Realize that some are toxic to birds, some are toxic to fish, bees. This will tell you what handlers need to wear, your workers if they go back in during the re-entry period. Know all that stuff. Realize that efficacy is not what EPA is considering when they're registering pesticides. They're considering whether or not you can protect yourself or non-target effects of these products, including organic ones. So just because it's labeled for a given disease doesn't mean it's proven effective for that disease. There's places on the web where you can get information, eOrganic. Plant Disease Management Reports has a lot. That's where a lot of us who are evaluating products post stuff. IR4. There's a resource guide that's come out of Cornell. It's available. As I said, I've got a web page where I'm posting a lot of stuff on organic management. Biofumigant mustard cover crops I've been looking at for managing some of our soil-borne pathogens. Need to get them into the ground. There's the debris incorporated. I've been looking at it for Phytophthora Blight, several things that they're effective for. We can talk about it tomorrow. We do have our discussion session tomorrow. So there's Phytophthora Blight. And the last one I wanted to mention is reduced tillage for managing some of our soil-borne diseases. I've been doing research on this for a number of years looking at conventional systems. I'm now part of a project that just started looking at it in organic systems. And we're eager and interested in working with growers who want to look at reduced tillage on their farm. This is an interesting project in, as the title says, it's farmer designed. So actually right now as we're meeting here today, we've got a group of organic farmers who have done reduced tillage for a couple of years, who have come together and are talking about how they do their systems and developing, pulling together their knowledge base and developing and designing programs and things that we need to work with. So if you're interested in reduced tillage, drop me an email, let me know. We're looking for more growers to be involved or hear about what we learned for the next 2-3 years, and then if things are going really well, you might want to jump into it. But we're looking at the reduced till, as I showed here, and we're also looking at permanent bed systems, what growers are doing now.
How about that, huh? Did you ever get the question?